All right, so let's talk about genetic engineering. What does it mean to engineer, to be an engineer? They design and build things. So genetic engineering is designing and building genetics. So genetic engineering is anything where you are changing something's DNA. So that cloning we talked about before, that is an example of genetic engineering. So if you change something's DNA, or maybe you transfer it from one organism to another, cutting genes out of so let me show you by example what I mean. All right, so let's say, for example, I need some insulin. I've got some patients with diabetes and I need some insulin and I can't get enough from pigs because that's how we used to get insulin. So I would like to train some bacteria to make insulin for me. So that's exactly what I'm going to do. So I'm going to need that part of the DNA. So I am going to cut the insulin part of my DNA, the little part of my DNA that codes for how to make insulin. And I need to go in, I need to cut it out. So I'm going to use these little restriction enzymes. We do this lab where we like cut with scissors and we say that we're cutting with restriction enzymes. And we cut out the insulin gene. And then I want to put that insulin gene into some bacteria to train that bacteria to make insulin for me. Wouldn't that be great? So that's what I do. So this is my bacterial DNA, and you'll notice it doesn't look like ours. It's not a long intertwining alpha helix chain or twisting alpha helix chain. It is actually a little circle. So bacterial DNA is a circle, and we call it a plasmid. So what we're going to do is we're going to take that circular DNA and we're going to chop a section out of it. We are going to cut it open. And I'm going to take my human insulin DNA and I'm going to insert it right in that spot. So what I have as a result, when I put it all back together, and because that's like the recombined DNA, we call it the recombined plasmid or recombinant DNA, I now have this new bacteria, this plasmid, which has the gene for making insulin in it. So what I've now made is a bacteria that can make insulin for me. And then another great thing about bacteria is they produce like crazy. We've got billions of them all over our bodies and all over everything we touch. Most of them are good or not bad for us. So all I have to do is just make one bacteria that's really good at making insulin, and then it's just going to reproduce like crazy. And then I'll have a whole bunch of bacteria that are really good at making insulin. And now I have synthetic insulin that I genetically engineered bacteria to make for me. So the process was let me go get some genes like the insulin gene that I need. I'm going to use restriction enzymes to cut them out of the human DNA. Okay, and then I am going to take that little piece of DNA, that insulin piece of DNA, and I'm going to insert it into the bacteria. And now I've made a bacteria that can produce the desired protein that they need because I put that DNA in there. And in this case, the protein that I needed was insulin. We can also use it for other things. So as I said, we've used this technique to have bacteria make insulin for us. We've also used it to make human growth hormone. That's HGH. If you put the human out in front of it, HGH. That is the hormone that, no, not right now, that like athletes illegally use to like, it's like performance enhancing. They're literally using human growth hormone to make themselves bigger. But it's also, we use it like illegally to help with like people who don't grow enough. We inject them with human growth hormone. Another example of how we've used this for good is we actually used guinea pigs. We genetically engineered them so that they would make clotting factors for people who have hemophilia. People with hemophilia, their blood doesn't clot, so they can't do like any activity because they could like bleed to death from like a little cut because their blood won't clot and stop bleeding. So we've used guinea pigs to create that human clotting factor for us, again, by cutting out the DNA that we wanted out of our own DNA and inserting it into their DNA. We've also used it for farming. So you've probably heard of GMOs or genetically modified organisms. You might have seen a label on something that said like no GMOs or non-GMO or but otherwise it pretty much, you can guarantee that your food does have GMOs in it. But all it really means is it's genetically modified organisms. So like I think 80% of all the corn in the US is all genetically modified. And what they've done, why would you want to genetically engineer your food? Well, you might want it to be resistant to destructive insects or fungal infections. So you have tomatoes that don't like get like nasty, like eaten by different insects or funguses. You might then not have to spray them with toxic insecticides, which could be a good thing. The only downside, and I don't know, maybe there's other downsides that I don't know about yet, but so what can happen is sometimes what they use it for is they make the crops so that, okay, let's say this is wheat. It is not hurt by insecticides. So they make it like anti-insecticides. Like, so then when you spray insecticides all over it, only the little insects and only the bugs die and not the plant. So that's good because you can just spray insecticide all over it, only kills the bugs, not the plants. Okay, so you've genetically engineered it to be able to survive insecticide or some other chemicals. But then the problem is now they can just spray the heck out of our crops, out of our food, because they're like, oh, it only hurts the bugs. Our plants are resistant to it. So for me, that's like the biggest downside that I've ever seen. You can watch different videos on whether GMOs are good or bad, but that you could result in like major spraying of chemicals onto our food because we've made our crops resistant to those chemicals, but now they're on our food. So just to conclude, genetic engineering, anytime we engineer, we plan and design and change the DNA of an organism or take the DNA of an organism, put another organism, that is genetic engineering. We have also used genetic engineering for different things in medicine. If you want to watch this gene therapy for Parkinson's, there's also other ones that are going to come up in our videos next week. So I'm not going to talk about them now, but you can watch that video.